Hello and welcome to Dr. Varsha Gaitonde's channel. In this video, we shall discuss some of the current affairs of GPB, some MCQs, and some general topics. The first topic is Kala Namak rice is now small, strong. This article appeared in the Hindu newspaper recently. Before getting into the actual topic, you should know what is Kala Namak rice, what is its importance, and all. As per the history, it is originated in the Buddhist era more than 2500 years ago. Now it is majorly cultivated in the regions of Uttar Pradesh, many of the districts. This is having typical aroma. Here you can see specialty rice of the world is the title to Kala Namak rice given by FAO, that is Food and Agriculture Organization. For its uniqueness, in the year 2013, this got GI, means Geographical Indication Tag, by Government of India. Kala Namak is said to outshine even the world famous basmati rice in all its quality traits and aroma, which is stronger than other basmati varieties. The typical and most important characters are its elongation, softness, cooking are the most important qualities. So, the typical advantages of eating or consuming Kala Namak variety of rice is this is rich in micronutrients like iron and zinc. Regular consumption prevents diseases like anemia as it is rich in iron, then seborrheic, alopecia, etc. Rich in antioxidants which help in preventing heart disease, that means for hair problem, heart disease, anemic condition, blood pressure and blood related problems. Then it also prevents Alzheimer's disease and more palatable and digestible. These are the typical characters. Along with that, this is having around 11 mg or 11% of protein content, which is more than the normal rice varieties. So this was all about Kala Namak. So now the scientists of IARI, those were collaborated with Acharya Narendra Dev University of Agriculture and Technology, which is located at Ayodhya. Previously, it was called as Faizabad. These scientists tested the two varieties, namely Pusa Narendra Kala Namak 1638 and Pusa Narendra Kala Namak 1652. Why these are in news? Because as we discussed, Kala Namak is having all the typical characters and good characters, but lodging is the major problem. Due to lodging, there is more chances of losing the harvest up to 50%. So, due to its dwarfness, the yield has doubled, and this has also got some of the other characters. This is all about Kala Namak rice. Now, when we are comparing Kala Namak for aroma and other characters with Basmati rice, we can see some of the recently developed Basmati rice varieties by marker assisted selection. One such variety is Pusa Basmati 1847, which is mass derived near isogenic line of a popular short duration Basmati rice variety Pusa Basmati 1509. That means Pusa Basmati 1847 is the progeny of Pusa Basmati 1509. The typical characters are resistant to both bacterial blight and blast. As you know, blast and blight are the two major diseases which reduces the yield in rice. Avoid the use of antibiotics and fungicides in the management of two major diseases, bacterial blight and blast. Yield is about 5.7 tons per hectare. Nursery days is 10 centimeter. Sowing date 21 days. Then this is recommended for Delhi, Punjab, Western Uttar Pradesh. Second Basmati kind variety is Pusa Basmati 1885. This is also mass derived and near isogenic line or nil of popular basmati rice variety Pusa basmati 1121. That means Pusa basmati 1885 is the progeny of Pusa basmati 1121, or derived line, you can say, resistant to again to bacterial blight and blast. This also avoids the use of antibiotics and pesticides in the management. This is again recommended for Delhi, then Punjab and Haryana. This gives yield of about 4.68 tons per hectare, which is somewhat lesser than this one, Pusa Basmati 1847. Then Pusa Basmati 1886, which is again a mass derived near isogenic line derived from Pusa Basmati 6. Again, the same characters and yield is almost on par with this 1885. This is recommended for cultivation in Haryana and Uttarakhand. Now we shall discuss one important tomato variety, that is Pusa golden cherry tomato 2. Now why tomato came here? Because as you know, biofortification became an important concept these days, and if anything is having high nutrient content means that will be brought into consideration. So the cherry tomatoes are different group of tomatoes that are mostly used raw in salad. This is very good for sambar purpose also, if we consider in South Indian conditions. Pusa golden cherry tomato 2 is the first indigenous golden yellow cherry tomato suitable and proposed for naturally ventilated polyhouse or low cost protected cultivation. As you know, when we expect more yield from tomato, either we will recommend it for protected cultivation or under the some specific conditions in field only. The variety has been developed by ICAR New Delhi, IAR we can say. Next article is about Russia Ukraine conflict hits sunflower oil imports. Why I am discussing here, as it is not much relevant to us, but as you know, during recent years, due to climatic vagaries and all, our farmers are giving less concentration towards the oil seeds as comparing to cereals and pulses. Even pulses are also majorly neglected if we compare it with cereals. Here, sunflower oil imports are hit by Russia Ukraine war. This means in India there is ample scope for sunflower cultivation. India imports 25 lakh tons of sunflower oil annually, which is 70% comes from Ukraine and 20% from Russia. So, when the imports are decreased means our farmers can go for more good kind of varieties so that our yield and oil production should increase. So, about the biology of sunflower, we shall discuss in our MCQ question discussion. Now, next topic is crop SR, that is a new tool for speeding up genetic research in crops. This is a tool or software tool for CRISPR research. This is created by Center for Advanced Bioenergy and Bioproduct Innovation, that is CABI, that is Department of Energy funded Bioenergy Research Center. Crop SR is the first open source software tool for genome wide design and evaluation of guide RNA or gRNA sequence for CRISPR research. As you know, CRISPR is one of the improved version of biotechnology, and this is somewhat different from our transgenic approach also. Here I am giving you an assignment. You should give the full form of CRISPR as well as crop SR in the comment section. Okay, so our next topic of discussion is Madhuban Gajar. This is again a biofortified variety which is benefiting more than 150 local farmers in Junagadh. It is planted in the area of about 200 hectares in Gujarat. This gives 40 to 50 tons per hectare, which has become the main source of income to the local farmers. It is used for various value added products like carrot chips, juice, pickle. This has a highly nutritious, developed through the selection method with higher beta carotene content of 277.75 mg per kg source of vitamin A and iron content of 276.7 mg per kg. This is developed by Sri Vallabhai Vasrambhai Marwania, a farmer scientist from Junagadh district. This variety is very important because generally in India, the major varieties or the varieties in limelight are developed by our scientists of agriculture, but here the Madhuban Gajar is developed by a farmer. This is confirmed with the national award by President of India at Rashtrapati Bhavan for during Festive of Innovation in 2017, and also conferred with Padma Shri in the year 2019 for his extraordinary work, and this was tested by National Innovation Foundation. It found that Gajar carrot variety possesses significantly higher root yield of 74.2 tons per hectare and plant biomass of about 275 gram per plant. This is cultivated more than 1,000 hectares of Gujarat, Maharashtra, Rajasthan, West Bengal, and Uttar Pradesh. Now we shall discuss some of the important revolutions. Before that, some more topics are there. In that, TNAU released new rice variety for sodic soil condition. As you know, the salt concentration will be more under sodic soil condition. This variety developed by Ambil Dharmalingam Agriculture College and Research Institute at Tiruchi. The new short duration variety Tri 5 was determined to be superior to Tri 2, the college's ex early duration variety appropriate for salt damaged soil, which was released in 2002. Similarly, Tri 3 and Tri 4, which were medium duration varieties, which were also released by the same college, that is Anbil Dharmalingam Agriculture College and Research Institute. Remember, this is for a specific condition for sodic soil situation. Two ethnic tribes of northeast wins the national award in agri sector. This is another topic. Under the supervision of Assam Agriculture University, two ethnic tribes from the northeast won two national awards in agriculture sector. This is for what? For Plant Genome Savior Community Award. That means Genome Savior. Savior means they conserve the varieties from a long period of time. This award is given to Dhonsuri Agri Farmer Cooperative Society of Kabri tribe and the Plant Genome Savior Farmer Recognition Award to Boloram Sarogsa of Dhimasa tribe. These are the two tribes from northeastern place which were recognized by Assam Agriculture University and got the Plant Genome Savior Award. So, now we shall discuss some of the revolutions and important years in agriculture. As you know, revolutions in India and their contributors I have already discussed in my previous video, but again I am bringing it to your notice because here, along with fathers of revolution, I have mentioned the period also. So, as you know, Green Revolution was contributed by M.S. Swaminathan during the year 1966 and 67. White Revolution for milk or dairy products by Varghese Kurian during 1970 to 1996. Blue Revolution is for fish and aquaculture products contributed by Dr. Arun Krishnan during 1973 to 2002. Golden Revolution is about fruits, honey, and horticulture crop improvement. That the father is Nirpak uh, Tutej, worked between 1991 to 2003. Silver revolution is for eggs, given by Dr. Mrs. Indira Gandhi, where she was Prime Minister of India during 1970s, and this silver revolution became popular in 2000s. Yellow revolution is about oil seeds. The father is Mr. Sam Patroda, worked during 1986 to 1990. Then pink revolution is about onion, prawns and pharmaceutical products. The father is Mr. Durgesh Patel during 1970s. Brown revolution is about cocoa and leather. Father is Mr. Harilal Chaudhary. Red level revolution is for meat and tomato. Father is Vishal Tiwari during 1980s. Golden fiber revolution is about jute, and it was popular during 1990s. Evergreen revolution is for overall production in agriculture. Again, Dr. Swaminathan is called as father of green and as well as evergreen revolution between 2014 to 2022. Black revolution is about petroleum. Silver fiber revolution is about cotton that became popular during 2000s. Round revolution is about potato between 1965 to 2005. Protein revolution is for higher agriculture production or for protein improvement. The term was coined by Mr. Narendra Modi, or the present Prime Minister of India, during 2014 to 2020. Then last one is grey revolution, is about fertilizers during 1960s and 70s. So for your knowledge, I have covered some more things here. Important years related to seeds. World Seed Year declared by FAO was 1961. As you know, National Seed Corporation established during 1963. Seed Act came in 1966. Seed rules during 1968. State Seed Farm Corporation came in 1969. Central Seed Certification Board during 1972. National Seed Program came during 1975 to 76. Seed Control Order 1983. National Seed Project 1979 to 80. New Policy on Seed Development 1988. National Seed Policy in 2002 and Seed Bill in 2004. So now we shall discuss some of the MCQs. First, we shall concentrate on plant breeding, then some questions related to genetics. The first question is progenitor of common bread wheat. As you know, there are many progenitors of common bread wheat. In that, there are four options, where Triticum monococcum, unknown species, and Triticum toshi. B species contributor is also found. You should keep it in comment section if you know. Here, all the above is the answer, because all have contributed as the progenitor of bread wheat, because it is a allohexaploid with AABB DD. This one, what genome composition. Second question is center of origin of oats is. The options are Near East, Mediterranean, Asia Minor, and all the above. The answer here is all the above, because in all these regions oat was found. Wild relatives of oats are. Options are Avena barbata, Avena fatua, both A and C, none of the above. Answer is both A and C. Here Avena barbata and Avena fatua both are the wild relatives of oats. The species contains genes for drought tolerance in barley are. Options are Hordeum spontaneum, Hordeum distichum, Hordeum intermedium, and all the above. The answer here is Hordeum spontaneum, which contains drought tolerance character. Next question is which of the following are the varieties of chickpea? Options are BDN 9-3, PUSA 256, BM4, and all the above. Answer is all the above because all the three are varieties of chickpeas only. Movement of sunflower head in the direction of sunlight from morning to evening is due to. As you know, sunflower, when sunrise happens, it will be towards east side. Then as the sun moves, it starts moving from east to west. Okay, like that, the phenomena is called as heliotropism. In sunflower, floret that bear seed is. Options are disc floret, ray floret, both A and B, and none of these. As when we were discussing about the oil exports imports to India, and I was telling about decreased import of sunflower due to Russia Ukraine war. I told that I'll discuss about the sunflower biology also. So, sunflower is almost a, often cross pollinated to cross pollinated crop. This is having a very fantastic or very bright kind of head consisting of disc floret and ray floret. For your better understanding, I'll just draw it here like this. This is the sunflower head, and here you will find ray and disc florets. Okay, the end one or the petal kind of formations which will be having only the rudimentary female kind of reproductive parts which are of not productive, that called as ray floret, and disc florets are the one which are hermaphrodite and having both male and female reproductive parts. So here the answer is disc floret. Okay, botanically the fruit of sunflower is called achene, grain, both A and B, and none of these. Answer here is achene. Sunflower is cross-pollinated crop due to protandry, self-incompatibility, both A and B, and none of these. Here the answer is both A and B because protandry as well as self-incompatibility characters are seen in sunflower. Okay. Next question is sunflower hybrids developed by using CGMS is or are. Options are BSH1, LDMRSH1, APSH11, all the above. Here the answer is all the above because all these varieties are developed by CGMS system only. Next question is safflower is which pollinated crop? Safflower will also be having same kind of flower condition, but the size of flower and the purpose. Here the main purpose of safflower flower during young means moderate condition is to extract the carthamin or the color compound, and also for the oil purpose, and this is often cross pollinated crop. Safflower is the often cross pollinated crop. Next question is Indian rapeseed, that is Brassica campestris, having three ecotypes are. इकोटाइप्स का मतलब होता है डिफरेंट क्लाइमेटिक कंडीशंस और हैबिटेट वेयर दे आर ग्राउंड. The options given here are Brassica campestris variety brown sarso, Brassica campestris variety yellow sarso, and Brassica campestris variety thoria. So here they have asked about the three ecotype. So the answer is all the above. Next is the Indian mustard Brassica juncea is the amphidiploid species between. First option is Brassica oleracea and Brassica campestris. Second is nigra and oleracea, Brassica nigra and campestris. As you know, if we'll draw the Brassica triangle, okay. So here you will see the Brassica nigra, then Brassica oleracea and Brassica rapa. Okay, in between these three you will find Brassica carinata, then Brassica napus, and Brassica juncea. So here Brassica juncea is asked. So the parents are Nigra and Repa. Okay, or you can say Nigra and Campestris. Repa is also called as Campestris. Okay, the mustard and rapeseed fruit botanically is three options: achene, capsule, silic, none of this. So here it is pod. Okay. The good quality attributes of mustard and rapeseed include: options are high erucic acid for industrial purpose, low erucic acid for edible purpose, low linolenic acid, and glucosinolate content, and all the above. The answer here is all the above, because when we consider rapeseed and mustard, for consumption purpose we need high for low erucic acid, then high erucic for industrial purpose, and low linolenic and glucosinolates for consumption purpose again, because these two are the toxic principles in rapeseed and mustard. Next question is Napier grass, that is Penicetum purpureum, is originated in the options are Near East, Himalayan region, Sub-Saharan tropical Africa, and none of this. The answer here is Sub-Saharan tropical Africa. Next question, the botanical name of forage bajra is Penicetum glaucum, Penicetum typhoidis, Penicetum americanum, and none of these. The answer here is Penicetum glaucum. Next and last question in plant breeding is useful fodder sorghum species are: the first option is Johansson grass, second Sudan grass, C both, then none. Here the answer is both, that is sorghum helipens as well as sorghum sudanis, both are for fodder purpose. Next, some of the cytogenetics and genetics questions. Cytoplasmic inheritance can be studied by: the options are backcross, test cross, top cross, and reciprocal cross. As you know, the definition of all of these and the purpose also. So, the cytoplasmic inheritance, which is obtained only from one parent, that is generally studied by reciprocal cross. So, the option is fourth one. Probes used in which molecular markers? There are many markers which are categorized as probe based and non probe used. Here AFLP and RAPD both are probe based molecular markers. So the answer is both B and C. Next question, DNA replication is unidirectional in: options are mitochondria, prokaryotes, eukaryotes, both A and B. As you know, mitochondria, chloroplast, and prokaryotes, all three contain unidirectional replication because they'll be having circular DNA. So here the option is both A and B. Answer. Fourth is the nucleosome histone octamer does not contain. As you know, histone octamer, there will be H2A, H2B, H3, H4, but H1 won't be there because it will be outside. So the answer is H1. Next is method not involving hybridization. Read this question carefully. Method not involving hybridization. Hybridization, the definition as you know, crossing of the two parents is nothing but hybridization, parent 1 and parent 2. Here the options given are molecular breeding, bulk method, pedigree method, and transgenic breeding. So when we consider molecular breeding, there we need this hybridization. In bulk and pedigree compulsorily we need, but in transgenic breeding, where we are concentrated on transferring the gene by external source, so we need not to go for a hybridization process. The answer is transgenic breeding. Sixth question is, in plant breeding, dash hybridization is extensively used. The options given are interspecific means the cross between species, intergeneric is cross between genera, intervarietal means between two varieties. Here you should concentrate on the gene pools. As you know, if we are going for two varietal cross, it will come under gene pool A, where the two are very compatible to cross. Here the answer is intervarietal, because the compatible crossing which comes between gene pool 1, whereas interspecific and intergeneric hybridization is quite unspecific or quite unsuccessful, and they come under gene pool 2 and 3 and so on. Okay, so the answer here is intervarietal. Next question is, among the seed propagated species, maximum mutant varieties have been developed in. The options given are wheat, barley, maize, and rice. As you know, wheat is self-pollinated, barley also, rice also, but maize is maximum cross-pollinated variety. So even in maize, there are self-mutant characters. What is that nature and character? You should give the details in the description box. So you study about the maize mutant nature, and the answer here is maize, where the maximum mutant varieties are developed under this crop. Next question, most suitable mapping population for identifying of gene or QTL in case of self incompatible and highly heterozygous species. Normally, if they'll ask, you may say that RILs or NILs, or majorly the RILs are used for studying the QTLs, but here the specification is given as self incompatible and highly heterozygous. When these two options are given, the answer is F1. Okay, here F2, RILs, NILs, and F1 are the option, and the answer is F1 population. Next question is, which one is not a restriction enzyme based DNA marker? Again, there are some restriction enzymes for cutting the edges are used in some of the markers like restriction fragment length, polymorphism, AFLP, and DART. But in SCAR, that is sequenced characterized amplified regions, here the restriction enzyme is not used. 10th question is, mRNA synthesis is catalyzed by. The options are RNA polymerase 1, 2, 3, and 4. The answer here is RNA polymerase 2. You should also know the functions of polymerase 1, 2, 1, 3, 4, and all. The next question is, mRNA usually is extracted using RNase. P is the answer here. Then GAA-TTC is a restriction sequence of CTT-AGG. This question is incomplete. So this is all about discussion of today. Thanks for watching. If you have any doubt, keep it under the comment section. Keep on subscribing, sharing, commenting. Thank you, friends.